everybody for being here today and for tuning into my presentation. I'm Jay Sharma, and the co-author to this paper is Dr. Vidhyacharan Bhaskar. And so our affiliation is with the Stanford University School of Medicine, but Dr. Vidhyacharan Bhaskar is also a professor at the University of San Francisco. And I am a high school student at Monte Vista High School. And my research is titled An RNA Sequencing Analysis of Glaucoma Genesis in Mice. So the objective of this research was to essentially find out more about glaucoma from a genetic standpoint. So there are 200,000 cases for glaucoma every year in the US alone. And in the people over the age of 60 years old, this is actually the leading cause of blindness. And although this is such a wide and prevalent problem all over the world, we don't know much about the genetic origins of glaucoma. So that's sort of what I wanted to, aim to discover in my project. So my project goal was to find a gene list of potential activator or protector genes for glaucoma. And essentially this means certain genes which could make it more likely for you to have glaucoma, or genes that make it more likely that you are protected from glaucoma. And so I did this using differential expression and gene ontology analyses. So I just wanted to go into a little bit about the biology behind glaucoma before I go into anything else. I'll cover the biology, and then I'll cover what other people have done in this field, and then I'll cover what my research is. So here is the picture of the eye, right? And you have the posterior chamber, and so your eye has aqueous humor in it, right? Which has these proteins and other things that are very essential for your eye. And so what happens is there's constantly aqueous humor flowing from the posterior chamber to the anterior chamber, and out of your eye and back into your body and being recycled. So this is always happening in your eye. And it essentially the aqueous humor will go from the posterior chamber into the anterior chamber and exit the eye through the trabecular meshwork. And so in glaucoma, what happens is that there is resistance to flow in this trabecular meshwork over here. And that leads to a high intraocular pressure, or IOP is the abbreviation for that. And this leads, so this higher pressure in the eye is what leads to glaucoma. And over time, the eye it's a degenerative disease, right? So it leads to cupping in the eye. And so what cupping means is that you can see this is the base of the eye, and there's the optic cup, and there's an optic disc. And so the ratio between these two things is very important to glaucoma. So if the ratio between these two things is greater than 0.5, then that's where you start to have glaucoma. The effects of glaucoma start to arise over there. And so for a normal person, it would be less than 0.5, but for someone with glaucoma, it might be higher. And so the certain kinds of symptoms that you have for glaucoma is that there's tunneling vision, as you can see over here, right? Your peripheral vision is not as good. You experience things such as headaches, right? And in order to track your glaucoma progress you need routine screening by an optometrist. And this is required because glaucoma is irreversible, right? So there is no cure for glaucoma known yet, but there are treatments. And there is routine screening that's required to make sure that your disease is not progressing too quickly and uncontrollably. And so glaucoma leads to an loss of peripheral vision, as I said before, which is also called tunnel vision. And you have fluctuating pain, headaches, blurred vision, halos surrounding the lights when you look at lights. And the symptoms are often worse at nighttime in the dark. If you're driving, then you might not be able to drive anymore during the night. And as a result, this affects many people on the personal level, which actually makes it a very important disease to tackle. So there are a few ways to diagnose this disease. One of which is non-contact tonometry. So non-contact tonometry is shown over here, where basically the machine will give a little puff to your eye, and the amount of pressure that's in your eye will be measured by the amount of air that comes back. So that's non-contact tonometry. The only problem with this method is that it's a little bit less accurate. The more accurate gold standard is actually called the Goldman applanation tonometry. And this actually takes a device that actually touches your eye and puts a little pressure on your eye in order to more accurately determine what is the pressure in your eye. So this is these two methods that I just explained were to determine the pressure that's in your eye. But you can actually also, look at the inside of your eye through this method called fundoscopy, as you can see over here, and that shows all the the blood vessels in your eye. And then you have this thing called the visual field assessment, which is less invasive, I would say, because you just look at a grid, and it tests your peripheral vision of what you're able to see that's outside of your line of sight. So these are the various ways that doctors will diagnose you with glaucoma. There are also certain treatments that are there right now. These treatments are actually only there to deal with the symptoms, like relieving the pain or reducing the symptoms, but they don't attack the problem at its root. So hopefully, out of the computational research that I've done for this project, future researchers can develop more effective treatments for glaucoma. And so the current glaucoma treatments, in order to understand these, you need to understand that this treatment has actually started when the pressure in your eye is above 24 mmHg. So I think a normal person would have around 21 or something like that. But if it becomes more than 24, then they would start the treatment for your eye. And this treatment falls under two major categories. One is increasing the uveoscleral flow, which is the flow of the aqueous humor, like I was talking about before, which is done through treatments such as letinoprost and brimonidine. And the other type of treatment is reducing the production of this aqueous humor. So that reduces the pressure in the eye and reduces the buildup of aqueous humor in your eye. And so that is done through timolol and durzolamide. And so certain risk factors for glaucoma include increasing age. As you get older, you're more likely to experience glaucoma. And if you have family history, then you would be more likely to have glaucoma yourself. And then Black ethnic origin also has a higher chance of having glaucoma, and nearsightedness or myopia is a risk factor for glaucoma as well. So here is a small literature review that I've laid out over here. You can see more of the literature review in my paper, but as for this, I've sort of summarized what the literature review is. So there's one paper by Chen et al. Which focuses on the time series for these different types of RNAs, right, and expression profiles for developing mice retina samples. And then the goal of this research was to find the key functional ncRNA and ceRNA which regulated this retinal neurogenesis. And so this is sort of similar to my research in some sense. And then Zhou et al. Basically aimed to identify the differing biological mechanisms that are involved in each glaucoma subtype. So you have they found that the high tension that you find in glaucoma patients had a correlation with the increased enrichment of genes associated with unfolded protein response. So that was their major finding in that paper. And Howell et al. Investigated the genetic origins of glaucoma using temporally ordered sequences of glaucoma states. And they found that a mutation in the C1qa gene, they found that those samples were actually protected from glaucoma, which is very interesting. However, so current research has not explored the stratification based on age and strain. And and so we lack insight into how these features, age and strain, contribute to and protect from glaucoma. So that's sort of what my research aims to show. So basically, it's a stratification based on age and strain, and performing these like pairwise experiments, genetic experiments, and computational genetic experiments to find out how these factors play a role in glaucoma, and basically finding gene lists for a certain protector or activator genes for glaucoma. So the data that I used was openly available from the Jackson Laboratory, and so there's the data samples themselves, and then there are two control groups. So for the data samples themselves, they're classified into no glaucoma or early 1, so that basically means kind of no glaucoma, and then no or early 2. That's basically similar to number 1, and then moderate glaucoma, as well as severe and severe glaucoma. So these are the different types of severity for glaucoma. And you have both optical nerve head and retina data provided for this these samples. And the one thing to note over here is that you want to compare apples to apples, right? So you want to compare on a genetic level. You want to compare optical nerve head data, perhaps to a control group with optical nerve head data. So you don't want to compare any data that is optical nerve head and retina data, because that would be like comparing apples to oranges. It's a little bit harder to understand what results you have. As for my control groups, I have two control groups, and like I said, like I have the stratification based on age and based on strain. So in the first control group, you have RNA sequence samples from the same type of mice. Different type of mice. So this is a stratification based on strain. So this strain of mice, D2 GPN MB plus mice, are of the same age as the DBA2J samples, which I just showed you over here, except they end up not getting glaucoma, which is very interesting. That this entire strain is basically a non-glaucoma control. And for this control group, you have both the optical nerve head and the retina data. So I'll detail in the next slide what kind of experiments I did, and like which pairwise experiments I set up in my experimental design. As for control group 2, so control group 1 was stratified based on strain, and control group 2 is stratified based on age. So you have mice that are from the same strain, but they're actually 4.5 months of age, which makes them pre-glaucoma. They're too young to have glaucoma. And if you analyze the difference between the RNA expression between, for example, severe DBA2J samples that are 10.5 months of age versus 4.5 months of age, you might find there are different RNA expression profiles. And based on this, you can create a list of potential activator or protector genes, which is what I did. And similarly, you can compare these severe samples to the other control group, which is the different strain. So before I went into any of my experimental design or any experimental design or comparisons, I did quality control. So I did a PCA visualization, principal component analysis, in order to just visualize my data and extract some very surface level meaning from my data, just visualizing. And then I did a dispersion visualization, so to identify the biological coefficient of variation to determine how reliable the data might be. And then I found the skewed genes in the data, which could actually identify which genes could serve as marker genes for the data. So after I did the quality control, I went on to my experimental design, which was finding differentially expressed genes, based on these 5 experiments that I have listed on the slide. So what I have is basically the severe optical nerve head versus the first control group. and then severe optical nerve head versus second control group. And both of these are optical nerve head data. You have severe optical nerve head versus no or early. Remember, this is the group that actually doesn't end up getting glaucoma. So you're always wanting to compare, mice with glaucoma with mice without glaucoma, mice with glaucoma, mice without glaucoma. And then the same thing over here, experiments 4 and 5 do a similar thing like 1 and 3, except they use the retina data as opposed to the optical nerve head data. So remember how I said comparing apples to apples? So this is what we're doing over here. So you can, for each of these experiments, what I did is I found the differentially expressed genes using differential expression analysis, after which I made a heat map to compare the gene expression and visualize the differentially expressed genes. And then I also made box plots to show how, show the expression of the differentially expressed genes and see how they vary across groups. And then for the differentially expressed genes, I took the top 1,000 differentially expressed genes, and then for both upregulated and downregulated genes, and I did gene ontology analysis on them and found the gene ontologies which correspond to these differentially expressed genes. And I'll go into what each of these mean and what they tell us in the next slides. So here are the results from the quality control. What I did is I visualized, on the one hand, I visualized the optical nerve head data samples and then the retina data samples. So there are two different graphs here. One graph is talking about optical nerve head data, and it shows the control groups over here. They're the control groups as well as the groups that do not have glaucoma are mostly clustered over here, whereas the moderate and severe groups are mostly clustered over here. And what does this tell us? This tells us that there might be some genetic, there's a very high chance that there is a genetic difference between these two groups, that it is actually worth exploring for the rest of this project. And although we can see some kind of separation, bear in mind that this is actually principal component analysis, which is a dimensionality reduction technique in linear algebra. And that we're not looking at the entire scope of the data. We're not looking at all the nuances that are visible in the data. So this is not entirely representative of our data, but it gives us a good idea that there are some kinds of genetic differences between mice with and without glaucoma. Over here, you can see the same thing for the retina data. You can see that the mice without glaucoma are over here, and then the moderate and severe groups are also over here. So there is a stark comparison. So the takeaway is that there might be genetic differences and that it's worth pursuing for the rest of this project. So the next thing is quality control for dispersion visualization. And so the dispersion visualization essentially will show us on the x-axis, it shows us the average log counts per million for a certain gene. And on the y-axis, you have the biological coefficient of variation for that gene across all samples. And each dot over here on this slide represents one gene. And so we have about 40,000 genes or so approximately that are plotted over here. And the overall trend is that the common biological coefficient of variation is about 0.278. And the industrial standard is that if it's less than 0.3, then that means it's good data. So we're allowed to proceed from here. Another thing I did for the quality control aspect, or the EDA part of it, is that I found the skewed genes, all the genes which had a high enough amount of skewness. And what I mean by that is I used Pearson mode skewness and basically found all genes which had an expression across samples which had a skewness of greater than 2 or less than -2. And overall, in the data set of 40,000 genes approximately, I found 112 skewed genes. And the takeaway for this is that these genes can serve as potential marker genes for different glaucoma groups. So for example, if I have an upregulation of this certain gene, gene A, then I might have, I might be placed into the category for severe glaucoma or something like that, or I might be at risk for something like that. So this will give us insight as well. As for the 5 pairwise comparisons, I've shown a sample for one of the pairwise comparisons, which I believe was the first pairwise comparison I showed you a few slides ago. But bear in mind that there are 5 of these experiments that are going on, and I can't show them all over here. But I think in my paper I've linked the GitHub where you're able to see all of the different results, and the tables are all there in the paper, I believe. So here you can see the between the severe group and the first control group for the optical nerve head data. You can see the different upregulated and downregulated differentially expressed genes. Over here in this table, specifically, I think I only have the upregulated genes, but you can see that there's going to be another table that I have in my paper that there is for the downregulated genes. And so the takeaway is that future researchers can look at these gene lists and use these gene lists to target to develop new glaucoma treatments. If you're able to inhibit a certain gene or somehow activate a certain gene, then like we could actually lead to genetic therapies or other things like that for glaucoma, which would be very helpful. Okay, so over here you can see the heat map for the top 20 differentially expressed genes. So I believe this is for the top 10 different upregulated genes and top 10 downregulated genes, so total 20 genes. And over here you can see the different genes, and the red and blue signifies the up and right down regulation. Excuse me. So the takeaway is that this is a good way to visualize the expression of the differentially expressed genes. Sorry, you can see the severe group here and then the control group here. And then finally, I did the gene ontology analysis, which is first the biological processes and then the cellular components and the molecular functions. And so you can see the different processes which are involved and which are relevant to the differentially expressed genes. So these tell us where, like what sort of processes that these genes contribute to within the cell. And that gives us more insight as to how do these genes play a role together within the cell and how could they contribute to glaucoma. As for this discussion, so I have the quality control results were good, that we were able to visualize the data and the variation in the data. So that was good. And the BCV was 0.278, which meant that it was good data. And I found 112 skewed genes, which could be marker genes for different types of severity for glaucoma. There is also the 5 pairwise comparison results, the experimental design I showed a few slides ago. And I was able to generate the gene list of potential activator or protector genes, which could be studied by future researchers to develop more treatments for glaucoma, more effective treatments. And then I visualized the differential expression through heat maps, as I showed on the previous slide. And that shows, the stark comparison between the upregulation and downregulation. And then I interpreted these gene lists using gene ontology, which would confirms the facts about glaucoma. Specifically, like the gene ontology, some of the gene ontologies tell us that this is indeed glaucoma because it involves neural processes, and but some other gene ontologies tell us what we don't know, and those are some gene ontologies which we can use and target in potential treatments. The shortcomings for this research are that the gene lists of upregulated genes in total are about greater than 1,000 genes large. The only thing is that I've sort of filtered based on like how important so like how the p-value or the log fold chain value and the other things. But there are over 1,000 potential genes that could serve as to be in the gene list or could be targeted in our research. And it's hard to decipher. It's hard to decipher these large gene lists, but I've sort of helped do that with the gene ontology analyses. But we still need to study these genes closely in order to find the marker, activator, and inhibitor genes, which is sort of what I've left for future research. And future areas for improvement for this project include collecting more RNA sequence samples, possibly from humans, and inhibiting. Another thing is inhibiting or activating certain genes in perhaps mice to see the effect on glaucomagenesis and develop more effective treatments, possibly gene therapy treatments. So a summary for my research is that, first I did the quality control insights, which were, that there is likely a genetic difference between the glaucoma and the control samples. And there is probably a stark one because we were able to see all of the control groups on one side and all of the glaucoma groups on the other side. And another thing is that the 5 pairwise comparisons, from that you can see that there are a lot of genes that are generated in these gene lists for these different experiments. But the good thing is that across these 5 pairwise experiments, the genes that tended to come up were very similar across gene lists, which reinforces our findings. And these gene ontologies confirmed known facts about glaucoma, so that serves to validate the research. The next steps for the research would be to increase the scope of the study. So basically, future researchers, like I was saying, can go through the gene lists and in depth and sort of find a potential activator or protector genes based on the filtering that I've essentially provided. And the gene lists can possibly be targeted in future research, like I was talking about, activating or inhibiting certain genes for starting with like mice or something like that, and then also using different kinds of data, like single cell data, which is very prevalent now, because this is bulk data, so using single cell data would be a huge improvement, and human RNA-seq data if it's attainable. So those would be good steps for future research. The lessons learned from this project, are that setting up an experimental design is very important, and that it's important to confirm the findings across multiple experiments, like I did. And the data quality is initially looking for the data quality, and the data control is quality control is very important in order to validate your findings. So these are the acknowledgements and my references over here. At this point, I'd like to thank you for listening to my presentation, and I would like to ask if anybody in the audience has any questions.